can let me know if my screen is visible and if I'm audible. Okay. So, good evening, everyone. Today is the 12th live session of this series. So, as you know that in these live sessions, what we do is that we basically discuss questions regarding the course RNA Biology, some previous year assignment questions, and we not only discuss the questions or the answers, we discuss the concepts behind these questions, the basic concepts behind these questions, so that it gets easier for us to solve similar type of other questions, right? So today is the 12th session of this course. So let's start by solving the first question for today. Okay. So we try to keep the sessions as interactive as possible. So if you know the answer, I'll wait for a few minutes anyway. But if you know the answer, you can just raise your hand or unmute and let me know. And we'll proceed accordingly. So the first question for today is this. mRNA capping ensure protection, translation, splicing, or all of the above? Which one do you think is correct? I'll wait for a few minutes. Is there any issue with the... Okay. Yeah. So the answer will be all of the above basically. So what is mRNA capping? We'll discuss about mRNA capping right now. Okay. So basically we know that the process of translation requires the process of transcription requires a lot of process like initiation, elongation, termination, etc. And there is a separate segment where we call mRNA processing in eukaryotes, which involves a 5' cap, a poly A tail, splicing out introns, and all of that. So what happens in the initiation phase is there are special promoter sequences with -10 and -35 nucleotide phases, and the sigma factor binds to the promoter, then the helices fit into the major groove, And in eukaryotes, there are several transcription factors, TF2, A, B, C, etc., that assemble and bind to promoter. There are additional sequences upstream, the CAAT box, enhancers, and silencers. Now, how does the regulation of transcription happen? There are cis and trans regulators. So there are cis factors, sequences upstream. So we have the Tata box enhancers, and there are trans factors, proteins that bind to promoter, made by far away genes. So what happens? Chromatin structure also regulates transcription. So the histones prevent transcription factors from binding. In interphase, DNA is in 30 nanometer fiber phase wound around the nucleosomes. So in elongation, what happens? The structure of RNA called 2. Okay. In eukaryote, special termination sequence, enzyme cuts transcript and adds a long string of A, poly A. Right? That is how it happens. And there are critical sequences that are in the introns, 5' U branch point A and 3' AG. Okay, so details of intron removal from pre-mRNA molecule. There is a cleavage at 5' exon intron junction and a formation of lariat structure. This is a branch point sequence. This is a lariat structure. There is a cleavage at 3' exon intron sequence junction and ligation of the two exons. There is a branch junction structure involving unusual 2' 5' phosphodiester bond between the G nucleotide from 5' junction and the branch point sequence. Okay. Now splicing checkpoints. Both the 5' splice site and the branch site are checked before any splicing reactions occur. The sequence must pass both identity checks. Okay. Now we have talked about the splicing mechanism, but in some of the previous classes, but very briefly, U2 and U6 hold the pre-mRNA in a way that juxtaposes the 5' SS and the branch point. The cleavage at the 5' SS and the addition of the U5 bring the 3' and 5' splice sites together. Now, what do we call splicing enhancers? There also may be special splicing enhancer sequences in the exons. Proteins and RNA bind to these sites and define the exons. Okay, so this is adapted from the main paper. If you can see this paper, T.D. Pollard and W.C. Earnshaw, Cell Biology. SNRNP, SR proteins and splicing factors associated with phosphorylation CTT or cytoplasmic CTT or C-terminal domain. Sorry. Now, splicing errors can lead to disease like spinal muscle atrophy, SMN1 and SMN2. Mutation in SMN2 causes a skipped exon in mRNA and all of that. Now, what are cell splicing introns? Group 1 introns are RNA, able to catalyze their own splicing reactions. Now, what's the first example of an RNA enzyme? Why does it matter? Important for ideas about the origin of life, often called the RNA world. Now, what about polymerase 1 and polymerase 2? Polymerase 1 transcribes ribosomal RNA. Ribosome is used to make proteins, has two subunits, large and small. Large subunit is composed of RNA 228S, 5.8S, 5S and many proteins. Small subunit has 18S RNA and many proteins. Now in eukaryotes, 18S, 5.8S and 28S RNA are adjacent with sequence repeating hundreds of times, transcribed as a unit and then cleaved. So RNA genes and RNA production in E. coli. What happens exactly? So in case of Pol 3 makes the tRNA and 5S rRNA. Transfer RNAs are necessary to make proteins. They bring the correct amino acid. Each tRNA has a unique sequence and slightly different shape. All are plus minus clover leaf. If we talk about transfer RNA, anticodon at one end recognizes mRNA structure sequence. Amino acid attachment site at the other, always CCA added after transcription and often have several modified bases, pseudouracil, ribothiamidine, etc. And these modifications happen post-transcription. It's a globally structure of each alanine tRNA. Now, the promoter for 5S and tRNA are within the gene sequence, not upstream. Now, this is a model for intron removal. This is a model for intron removal by the spliceosome. If you check, this is a branch point alanine, 3' splice junction, 5' splice junction. and U1 snRNP binds to 5' end of intron. U2 snRNP binds to the branch point, and U4, U6, and U5 snRNPs bind to U1 and U2, and the loop forms. U4 snRNP is released, active lysosome, mature mRNA, excised intron sequence in lariat shape, still complex with snRNPs. It's released intron RNA in lariat shape. Now, what do we call alternative? Some genes have alternative splicings that different exons are used in different tissues. Now, if you talk about RNA editing in some species, the final RNA molecule is much longer than the corresponding DNA sequence, right? Extra bases are added after transcription. Okay. Now, one example of this is spinal muscle atrophy, one of the most common genetic causes of childhood mortality. In humans, an essential gene called SMN has been duplicated or compared to other mammals such as mouse. SMN2 encodes a protein identical with SMN1, but it is expressed at much lower level because a silent mutation in one exon interferes with the binding of an SR protein, resulting in exon skipping in most but not all of the SMN2 mRNAs. The spinal muscle atrophy occurs when mutations occur in SMN1 that inactivate the gene. Sufficient SMN protein is expressed from SMN2 to allow embryonic and fetal development. However, during childhood, spinal cord neurons die because of insufficient SMN, resulting in disease. This is RNA ribonucleic acid. It's a polymer of ribonucleotides linked together when trying to 5' phosphodiester linkage. This RNA versus DNA, if you see over here. The difference between RNA and DNA is single-stranded mainly except when self-complementary sequences are there, it forms a double-stranded structure. DNA is double-stranded except for certain viral DNAs which is single-stranded. RNA ribose is the main sugar, and DNA the sugar moiety is deoxyribose. In RNA, pyrimidine components differ, thymine is never found except tRNA. In DNA, thymine is always there but inosine is never found. In RNA, being single-stranded structure does not follow Chargaff's rule. In DNA, it does follow Chargaff's rule. The total purine contained in double-stranded DNA is always equal to pyrimidine content. RNA can be easily destroyed by alkalis to cyclic diesters of mononucleotides. DNA resists alkali action due to the absence of OH group at 2' position. RNA is a relatively labile molecule, undergoes easy and spontaneous degradation. DNA is a stable molecule. Spontaneous degradation is very slow. Genetic information can be stored for years together without any change. Now, RNA is mainly cytoplasmic but also present in nucleus, primary transcript and small nuclear RNA. Whereas DNA is mainly found in nucleus, extranuclear DNA is found in mitochondria and plasmids, etc. The base content varies from 100 to 5000. Size is very billions of base pairs are there depending upon the organism. Now, in RNA, there are various types of RNA: mRNA, messenger RNA, ribosomal RNA, rRNA, tRNA, transfer RNA, snRNA, short nuclear RNA, siRNA, short interfering RNA, miRNA, microRNA, hnRNA. These RNAs perform different and specific functions, whereas DNA is always of one type and performs the function of storage and transfer of genetic information. In RNA, there is no variant but physiological forms of RNA found. The different types of RNA do not change their forms. However, in DNA, there are variable forms of DNA, physiological forms. In RNA is synthesized from DNA, it cannot form DNA except by the action of reverse transcriptase. Okay? And then also it's called cDNA or complementary DNA. It cannot duplicate except in certain viruses where it is a genomic material. Whereas in DNA, it can form DNA by replication, it can also form RNA by transcription. Many copies of RNA are present per cell, which is known as the transcriptome, whereas a single copy of DNA is present per cell. As we said, there are various types of RNA mRNA, tRNA, RNA, siRNA, small nuclear RNA, microRNA, small interfering RNA, heterogeneous nuclear RNA, hnRNA. Now, messenger RNA comprises only 5% of the RNA in the cell, most heterogeneous in size and base sequence. All members of the class function as messengers carrying the information in the gene to the protein synthesizing machinery. Structural characteristics of mRNA: the 5' terminal is capped by 7-methyl guanosine triphosphate cap. The cap is involved in the recognition of mRNA by the translating machinery. It stabilizes mRNA by protecting it from 5' exonuclease. Then we have the 3' end of most mRNAs are polymer of adenylyl residue. The tail prevents the attack by 3' exonucleases. Histones and interferons do not contain poly A tails. On both 5' and 3' end, there are non-coding sequences which are not translated. And the intervening region between non-coding sequences present between 5' and 3' end is called the coding region. This region encodes for the synthesis of a protein. Now, the 5' cap and the 3' tail impart stability to mRNA by protecting from specific exonucleases. So, this is the 5' cap over here and the poly A tail at the 3' end. Now, the mRNA molecules are formed with the help of DNA template leading towards the transcription. The sequence of the nucleotides in mRNA is complementary to the sequence of nucleotides on template DNA. The sequence carried on mRNA is styled in the form of codons. A codon is made up of 3 nucleotides. mRNA is formed after processing of heterogeneous nuclear RNA. In mammalian nuclei, hnRNA is the immediate product of gene transcription. The nuclear product is heterogeneous in size and is very large, and the molecular weight may be more than 10 to the power 7, while the molecular weight of mRNA is less than 2 to the power 6. 75% of hnRNA is degraded in the nucleus, only 25% is processed to mature mRNA. This is the hnRNA. The mature mRNA is formed from primary transcript by capturing, mapping, tailing, splicing, and base modification. Now, transfer RNA are the smallest of 3 major species of RNA molecules. They have various nucleotide residues. Synthesized by the nuclear processing of a precursor molecule, they transfer the amino acids from cytoplasm to the protein synthesizing machinery, hence the name tRNA. They are easily soluble, hence called soluble RNAs. They are also called adapter molecules since they act as adapters for the translation of the sequence of nucleotides of the mRNA into specific amino acids. There are at least 20 species of tRNA, one corresponding to each of the 20 amino acids required for protein synthesis. Now, structural characteristics of tRNA. Primary structure: the nucleotide sequence of all the tRNA molecules allow extensive intra-strand complementarity that generates a secondary structure. The secondary structure is each single tRNA shows extensive internal base pairing and acquires a clover leaf-like structure. The structure is stabilized by hydrogen bonding between the bases and is a constant feature. What are the structural characteristics of tRNA? All tRNAs contain 5 main arms or loops, which are as follows: acceptor arm, anticodon arm, reactive arm, and all of that. Now, secondary structure of tRNA: the acceptor arm is at 3' end. It has 7 base pairs. Their sequences and pairs: cytosine, RNA at 3' end. 3' OH group terminal of adenine binds with the carboxyl group of amino acids. The tRNA bound with amino acid is called amino acid tRNA, and the CC attachment is done post-transcriptionally. The carboxyl group of amino acid is attached to the 3' OH group of adenine nucleotide of the acceptor arm. The anticodon arm of base pairs with the codon present on the mRNA. If you talk about the anticodon arm, it lies at the opposite end of the acceptor arm, 5 base pairs long, recognizes the triplet codon present in the mRNA. Base sequence of the anticodon arm is complementary to the base sequence of mRNA codon. Due to the complementarity, it can bind specifically with mRNA by hydrogen bonds. The DHU arm, it has 3 to 4 base pairs as a recognition site for the enzyme that adds the amino acid to the acceptor arm, amino acid tRNA synthetase. Then there is a TYC arm. This arm is opposite to the DHU arm since it contains uridine, that is why it is so named. It is involved in the binding of tRNA to the ribosomes. There is an extra arm or variable arm. About 75% of tRNA molecules possess a short extra arm. If about 3 to 5 base pairs are present, the tRNA is said to be belonging to class 1. Majority tRNA belongs to class 1. The tRNA belonging to class 2 have long extra arm, 30 to 21 base pairs in length. The L-shaped tertiary structure is formed by the further folding of the clover leaf due to the hydrogen bonds between T and D arms. The base paired double helical stems can get arranged into double helical columns, continuous and perpendicular to one another. The mammalian ribosome contains two major nucleoprotein subunits, a larger one molecular weight of 2.8 × 10⁶ and a smaller subunit with a molecular weight of 1.4 × 10⁶. 68 subunit contains a 5S ribosomal RNA, a 5.8 srRNA, and a 28 sRNA. There are probably more than 50 specific polypeptides. The 40 subunit is smaller and contains a single 18 srRNA and approximately 30 distinct polypeptide chains. All of the ribosomal RNA molecules except the 5S rRNA are processed from a single 45S precursor RNA molecule in the nucleus. 5S rRNA is independently transcribed. So ribosomal RNA. Now the functions of ribosomal RNA molecules in the ribosomal particle are not fully understood, but they are necessary for ribosomal assembly and seem to play key roles in the binding of the mRNA to the ribosomes and its translation. Recent studies suggest that RNA component performs the peptidal transferase activity and thus is an enzyme. Now, small RNA, most of these molecules are complexed in wall proteins to form ribonucleoproteins and are distributed in the nucleus, cytoplasm, or in both. So they range in size from 20 to 300 nucleotides and are present in 100,000 or 1 million copies per cell. So we have talked about small nuclear RNAs, such as small RNAs significantly involved in the mRNA processing and gene regulation. Of the several snRNAs, U1, U2, U4 and U5 and U6 are involved in neutron removal and the processing of hnRNA into mRNA. E7 snRNA is involved in the production of the correct 3' ends of the histone mRNA which lacks the poly A tail. Small nuclear snRNAs are involved in the process of splicing or primary transcript to form mature mRNA. The snRNA complex forms complexes with proteins to form ribonucleoprotein particles called snRNPs. These two classes of RNAs represent a subset of small RNAs. Both play important roles in gene regulation. miRNAs and siRNAs cause inhibition of gene expression by decreasing specific protein production, albeit apparently via distinct mechanisms. Then we have microRNAs, which are typically 21-25 nucleotides in length and are generated by nucleolytic processing of the products of distinct genes or transcription units. The small process mature miRNAs typically hybridize by the formation of imperfect RNA duplets within the 3' untranslated regions of specific target mRNAs, leading by unknown mechanisms to translation arrest. If miRNAs, short non-coding RNAs present in all living organisms, have been shown to regulate the expression of at least half of all human genes. These single-stranded RNAs exert their regulatory action by binding messenger RNAs and preventing their translation into proteins. Then we have the small interfering siRNAs that react with specific nucleolytic cleavage of larger double-stranded RNAs to again form small 21-25 nucleotide long products. These short siRNAs usually form perfect RNA-RNA hybrids with their distinct targets potentially anywhere within the length of the mRNA where the complementary sequence exists. Formation of such RNA-RNA duplexes between siRNA and mRNA results in reduced specific protein production because the siRNA-mRNA complexes are degraded by dedicated nucleolytic machinery, and some of all of this mRNA degradation occurs in specific occurrence termed T-bodies. Right? What are small interfering RNAs? They are 20-25 nucleotides long, double-stranded RNA molecules that have a variety of roles in the cell. They are involved in RNA interference pathway that interferes with the expression of a specific gene by hybridizing creates corresponding RNA sequence. In the target mRNA, this then activates the degrading mRNA. Once the target mRNA is degraded, mRNA cannot be translated into protein. So what is the significance of mRNAs? Is that both mRNAs and siRNAs represent exciting new potential targets for therapeutic drug development in humans. In addition, siRNAs are frequently used to decrease or knock down specific protein levels in experimental procedures. In lab, extremely useful and powerful alternative to gene knockout technology. So RNA exists in several different single-stranded structures, most of which are directly and indirectly involved in protein synthesis and regulation. The linear array of nucleotides in RNA consists of A, G, C, U, and the sugar moiety is ribose. Major forms of RNA are already discussed, and RNA molecules act as catalysts like ribozymes. And mRNAs and siRNAs represent exciting new potential targets for therapeutic drug development in humans. Now, coming to the post-translational modification of RNA. So, primary transcript is the initial linear RNA copy for transcription unit, the segment of DNA between specific initiation and termination sequences. Now, the primary transcripts of both prokaryotic and eukaryotic tRNA and RNA are post-transcriptionally modified by cleavage of the original transcripts by ribonucleases. So, tRNAs are further modified to help give each species its unique identity. So, in contrast, prokaryotic mRNA is generally identical to its primary transcript, whereas eukaryotic mRNA is extensively modified both co and post transcriptionally. Right? So, RNAs of both prokaryotic and eukaryotic cells are generated from long precursor molecules called pre-RNAs. So, the 23S, 16S, and 5S rRNA of prokaryotes are produced from a single pre-RNA molecule, such as the 28S, 18S, and 5.8S rRNA of eukaryotes. The pre-rRNAs are cleaved by ribonucleases to yield immediate sized pieces of rRNA, which are further processed, trimmed by exonucleases, and modified at some bases and riboses to produce the required RNA species. So the post-transcriptional processing of eukaryotic ribosomal RNA by ribonucleases, RNases. The primary tRNA transcript, that is A, functional tRNA after post-transcriptional modification. Modified bases include dihydrouracil, C-diuracil, and M, which means that the base has been methylated. C, a 16 nucleotide sequence at the 5' end is cleaved by RNase P. A 14 nucleotides shown in the purple in the anticodonal loop is stimulated by splicing. Uracil residues at the 3' end are replaced by the CCA sequence found in all mature tRNAs, and many bases are converted to characteristic modified bases shown in yellow. Now, both eukaryotic and prokaryotic tRNA are also made from longer precursor molecules that must be modified. Sequences at both ends of the molecule are removed, and if present, an intron is removed from the anticodon loop by nucleases. Now, what is eukaryotic mRNA? The collection of all primary transcripts synthesized in the nucleus by RNA polymerase II is known as heterogeneous nuclear RNA. The pre-mRNA components of the hnRNA undergo extensive co- and post-translational modification in the nucleus. These modifications include 5' capping, like with 7-methyl-guanosine, 3' poly A tail addition, removal of introns, and alternative splicing of the mRNA molecules. Now, what is the 5' capping of 7-methyl-guanosine? So, the cap is a 7-methyl-guanosine attached backward to the 5' terminal of the mRNA, forming an unusual 5' triphosphate linkage. Now, creation of the cap requires removal of the gamma phosphate from the 5' triphosphate of the pre-mRNA, followed by addition of the GMP, guanine monophosphate, from GTP, guanine triphosphate, by the nuclear enzyme guanine transferase. Now, methylation of this terminal guanine occurs cytosol and is catalyzed by guanine 7-methyl transferase. Now, S-adenosyl methionine is a source of methyl group additional methylation state may also. So, the addition of this 7-methyl guanosine cap helps stabilize the mRNA and permits initiation of translation. Eukaryotic mRNA is lacking the cap and not efficiently translated. Now, if we talk about the poly A tail addition, most eukaryotic mRNA have a chain of 40 to 200 adenine nucleotides attached to the 3' end. This poly A tail is not translated from the DNA but rather is added after transcription by the nucleic enzyme polyadenylate polymerase using ATP as a substrate. So, the mRNA is cleaved downstream of a consensus sequence called the polyadenylation signal sequence AAU-AAA, found near the 3' end of the RNA, and the poly A tail is added to the new 3' end. These tails help stabilize the mRNA facilitate its exit from the nucleus and aid in translation. After the mRNA enters the cytosol, the poly A tail is gradually shortened. If we talk about the removal of introns, maturation of eukaryotic mRNA usually involves the removal of RNA sequences, introns and intervening sequences, and which do not code for protein from the primary transcripts. The remaining coding sequences, the exons, are joined together to form the mature mRNA. Now, the process of removing introns and joining exons is called splicing, and the molecular complex that accomplishes these tasks is known as the spliceosome. So, few eukaryotic primary transcripts contain no introns, for example, those from histone genes. Others contain a few introns, whereas some, such as the primary transcripts for the alpha chains of collagen, contain more than 50 intervening sequences that must be removed before mature mRNA is ready for translation. Okay, splicing is an RNP small nuclear ribonucleotide. This is the basic schematic. There can be alternative splicing of mRNA molecules. The pre-mRNA molecules from some genes can be spliced in alternative ways in different tissues, and this produces multiple variations of the pre-mRNA of the mRNA and therefore of its protein product. So this appears to be a mechanism for producing a diverse set of proteins from a limited set of genes. So alternative splicing is a diverse set of proteins from a small set of genes. You see over here. It can be exon 1, exon 2, exon 3 or exon 1 and exon 3. This is basically a summary of whatever we discussed. I have already talked about all of these. Need to go into too much detail. Already discussed 5' cap, poly A tail, splicing, editing, coupled with elongation, all of these. You see, this is the 5' Cas7 methyl guanosine, splicing reactions, and lariats, signal RNA splicing. All of we discussed RNA splicing mechanism, splicing errors, additional factors for correct splicing site recognition, RNA factory concept, exon definition, hypothesis, RNA factory, etc. Cell splicing, the poly A tail, and all of that we've already discussed. We have taken these from major molecular biology books, so you can just go and check these out. We've also discussed all of these. RNA is inherently unstable, especially at the ends. So the ends are modified to protect it. At the 5' end, a slightly modified guanine 7-methyl guanosine is attached backwards via 5' to 5' linkage to the triphosphates on the first transcript base. And at the 3' end, the primary transcript RNA is cut at a specific site and 100 to 200 RNA nucleotides are attached to poly A tail, so that the A's are not coded in the DNA of the gene. All of these have been discussed. All of these are taken from books. So coming back to the question that we started with, mRNA capping ensures protection, translation, splicing, all of the above. Okay. So this is the correct answer. I'll wait for a few minutes. If you have any doubt, you can ask me. Then we move on to the next question for today. Any doubts? Okay, so the next question, move on to the next question if there are no doubts in this question. Okay, so the next question for today is cellular senescence is seen in all cell types. Which one do you think is correct? True or false? Okay, I'll wait for a few minutes. You discuss the questions among yourself and try to answer. Then I'll move ahead with the answer. Okay. So for cellular senescence, it is false. It is not seen in all types of cells, all cell types. So if you talk about senescence or aging, so what does it basically mean? Aging is generally characterized by the declining ability to respond to stress, increasing homeostatic imbalance, and increased risk of aging-associated diseases. Right. So death is the ultimate consequence of aging. Now, differences in maximum lifespan between species corresponding to different rates of aging. For example, inherited differences in the rate of aging make a mouse elderly at 3 years and a human elderly at 90 years, right? Genetic differences affect physiological processes like efficiency of DNA repair, antioxidant enzymes, rates of free radical production, etc. If you talk about senescence, the process by which a cell loses its ability to divide, grow, and function, this loss of function ultimately ends in death. So it's a degenerative process only. This is only the process by which a cell loses its ability to divide, grow, and function. This loss of function ultimately ends in death. It's a degenerative process only. Has no positive features. Right? Traditionally, aging was explained in two theories. Program theories imply that aging is regulated by biological clocks operating throughout the lifespan. This regulation would depend on changes in gene expression that affect the systems responsible for maintenance, repair, and defense responses. Stochastic theories blame environmental impacts on living organisms that induce cumulative damage at the various levels as a cause of aging. Examples which range from damage to the deoxyribonucleic acid, damage to tissues and cells by oxygen radicals, and cross-linking. Now, more recently, theories of aging are categorized by as the molecular gene theory, coronal restriction, somatic mutation, gene regulation, or cellular theory, free radical theory, wear and tear theory, apoptosis, senescence, telomere loss, cellular stress, system theory, rate of living theory, neuroendocrine theory, immunologic theory, and the evolutionary theory, disposable soma, antagonistic pleiotrope, mutation accumulation. Now, if you talk about the molecular gene theories, fidelity accuracy of mRNA translation is impaired due to inability to decode codons in mRNA. Fidelity of gene expression declines with age, resulting in increased fraction of abnormal proteins. Accumulation of molecular damage, primarily to DNA genetic material. Disdifferentiation, agile accumulation of random molecular damage impairs regulation of gene expression, and the gene regulation. Aging caused by changes in gene expression, regulating both aging and development. Gene expression, protein folding, and activity. If we talk free radicals, so oxidative metabolism produces highly reactive free radicals and subsequently damage protein and DNA. Then we have the mitochondrial DNA damage, wear and tear, accumulation of normal injury, glycosylation theory of aging, products from glucose with proteins, oxidation, age, theory of aging, etc. Our process program cell death resulting from intrinsic damage and genetically determined events of genome crisis. Senescence phenotypes of aging are caused by an intense increase in frequency of senescent cells. Senescence may be the result of telomere loss or cell stress. Now, somatic cells usually lack telomerase activity, which means that telomeres shorten with each cell division. Now, cultured cells may go into crisis as a result of reaching zero telomere length. The reactivation of telomerase enables cells to survive crisis and to become immortal. Now, system theories. What is the rate of living? So, assumes a fixed amount of metabolic potential for every living organism. Live fast, die, and health, everything. Then the neuroendocrine. Alterations in neuroendocrine control of homeostasis results in the age-related physiological changes, also referred to as the neuroendocrine theories of aging. Then we have immunology. This decline of immune function with age results in increased incidence of disease, also referred to as the immunological theory of aging. What are the evolutionary theories? Like the disposable somatic cells are maintained only to ensure continued reproductive success. Following reproduction, soma is disposable. Theory. Here the antagonistic eutrophic genes that are beneficial at younger ages are deleterious at older ages. Here the mutation accumulation. Mutations that affect health at older ages are not selected against. Now, the free radical theory of aging, oxygen free radicals generated cause cumulative oxidative damage resulting in structural degeneration, functional decline, and age related diseases. Some believe that oxidative stress is the predominant cause of age associated degenerative change. Now, for mitochondrial dysfunction and aging and mitochondrial theory of aging, oxidative stress is going up, mitochondrial DNA damage is going up, mitochondrial DNA depletion, oxidized proteins, lipid peroxidation, lipid formation, increase in repair systems. Now, oxidative stress and aging. What happens when oxidant production is greater than antioxidant defenses? In oxidative stress, DNA damage, protein damage, lipid damage, all of these happens. Free radical theory, oxidative metabolism produces highly reactive free radicals that subsequently damage protein and DNA. Evidence from model organisms, superoxide dismutase transgenes can extend the lifespan of Drosophila. Chemicals that mimic catalase activity can extend C. elegans lifespans. Long-lived mutants are typically stress-resistant, including resistance to drugs. Lifespan extension by insulin-like signaling mutants in C. elegans requires catalase activity. Now, what is the inflammation theory of aging? The molecular inflammation theory of aging is based on specific anti-aging mechanism of calorie restriction, such as the reduction in TNF alpha NF kappa B signaling. The inflammation theory of aging goes hand in hand with the free radical theory of aging and the glycooxidation theory of aging. So, what are the hormone theories in declining estrogen? DHEA, dehydroepiandrosterone, is secreted by the adrenal glands and called a master hormone because it is converted into many other hormones involved in growth and strength. Supplements of DHEA, which declines with age in both men and women, have been shown in animal studies to reverse many debilitating signs of aging. The risks of cancer with long-term use and risk of prostate cancer, etc., are unknown. In general, humoral immunity declines with age, and the onset can occur as early as when an individual reaches sexual maturity. Now, aging appears to affect T cell number function. Decline could lead to increase in risk to viral infections and cancer. Cell loss and apoptosis, shift in the proportion of subpopulations, and the quantitative cellular changes have all been detected. Now, cross-linking sugar related to the aging process. Yes, this aging process is also governed by certain chemical molecules and certain formations that are likely to participate in reactions to slow you down, which are not intended by genes. Now, what is the apoptosis theory of aging? Apoptosis could be activated prematurely in most mitotic cells and contribute to the aging process. What is the mechanism with age and what mechanisms can lifelong exercise prevent age-related loss in post-mitotic cells? Okay, so the key causes is shortening of telomeres below critical length is with reduced longevity. Increase of P53 above wild type levels can decrease tumor formation but also decrease longevity. Now, what are therapy strategies to promote healthy aging and to prolong life? Interventions that promote health and prolong maximum lifespan, lifelong caloric restriction, lifelong exercises, anti-inflammatory compounds, antioxidant therapy, genetic engineering and organ transplant, etc. Now, what are summaries? Primary and secondary aging. Primary aging program changes in gene expression, wear and tear from reactive oxygen and nitrogen intermediates, lipid peroxidation products, and path like glycation end. Products influenced by inflammatory mediators. Okay. So, I have discussed all of these. We will not go into much detail. So, what is it? What causes it? So, response of normal cells to potentially cancer-causing events, exceptions with germ cells, early embryonic cells, many tumor cells. What happens when cells exhaust their replicative lifespan? So, irreversible arrest of cell proliferation, universal resistance to apoptosis in the stem cells, and altered function. Universal was cell type specific, senescent phenotype. Cell proliferation is equal to telomere shortening, DNA damage, oncogene expression, supermitogenic signals. What do inducers of the senescent phenotype have in common? cell proliferation, DNA damage, all of these oncogenes, strong mitogens can potentially cancer causing. So, cellulosis is an important tumor suppressive mechanism induced by potentially oncogenic events. Most tumor cells are immortal. Many oncogenes act by allowing cells to bypass the senescence response, and senescence is controlled by the two most important tumor suppressor genes, P53 and PRB. Mice with cells that do not die as a senescence die young of cancer. Now, cancer is caused by genetic mutations in epigenetic tissue environment events. Now, activation of oncogenes, inactivation of tumor suppressor genes. Inactivation of tumor suppressor genes encoding P53 and PRB proteins is most common. Now, P53, now nuclear proteins controlled by complex pathways, upstream regulators and downstream effectors. Control expression of other genes, halt cell cycle progression in response to induced senescence, and crucial for allowing normal cells to sense and respond to senescence signals. Senescent phenotype entails changes in cell function. Aging is a consequence of the declining force of natural selection with age. Now, what is the antagonistic pleiotropy? Some traits selected to optimize fitness in young organisms can have unselected deleterious effects in old organisms. Okay, so selected antagonistic pleiotropy is unnecessarily selective tumor suppression. Functional changes unselected deleterious. Functional changes associated with solution of molecules that can be detrimental to tissues if not controlled. Senescent fibroblasts secrete proteases, growth factors, inflammatory cytokines. Cells from old donors divide less often than cells from young donors. Cells from hot-lived species are more sensitive to senescence induces, particularly oxidative stress, than cells from long-lived species. Cells from donors with premature aging syndrome senescence more readily than cells from normal donors. And senescent cells expressing a senescence marker accumulate with age and at sites of age-related pathology. So, pseudocytosis is seen in all cell types. The answer is false. If you have any questions, I'll wait for a few minutes and then I'll move on to the next question for today. any problem with the. Okay. Okay. Going to the next question for today. Before participation in protein synthesis, the tRNA must get A. Modified, B. Amino acylated, C. Denatured, D. Transacylated. Which one do you think is the right answer? Okay. So the answer will be amino acylated, right? So if we talk about from the basic of molecular biology, we know the central dogma. Okay, maintenance of genes to gene replication, transcription of information from DNA to mRNA, translation of mRNA into protein. We know about the nucleic acids and polymers of nucleotides. This nucleotide includes a base that is either a purine or a pyrimidine, hydrogen or guanine, or a pyrimidine, cytosine, uracil, or thymine. Then we have some nucleic acids contain modified bases. Examples: nucleoside bases found in RNA, include modified bases found in tRNA.
So in nucleotide example, adenosine monophosphate, the base is bonded to ribose sugar, which is a phosphate in ester linkage to the 5' hydroxyl, right? Now nucleic acids have a backbone of alternating pi and ribose moieties. Phosphodiester linkages form as the 5' phosphate of one nucleotide forms an ester link with the 3' OH of the adjacent nucleotide. Now, hydrogen bonds linked to complementary nucleotide bases on separate nucleic acid strands on or complementary for the same strand. Conventional base pairs UOT and CNG. Okay. Now, base pairing over extended stretches of complementary base sequences into nucleic acid strands stabilizes secondary structures such as the double helix of DNA. Stacking interactions between adjacent hydrophobic bases contribute to stabilization of such secondary structures. Okay, and each base interacts with its neighbors above and below in the ladder-like arrangement of base pairs in the double helix. Now, genetic code is based on the sequence of bases in a longer genetic nucleic acid. Each codon or sequence of 3 bases in mRNA codes for a particular amino acid or for chain termination. Some amino acids are specified by 2 or more codons. Now, pseudonyms in most cases differ only in the third base. Similar codons tend to code for similar amino acids. These effects of mutation are minimized. Now, tRNA. The genetic code is read during translation by adapted molecules, tRNAs, that have 3 base anticodons complementary to codons in mRNA. Wobble reading of the mRNA allows some tRNAs to read multiple codons that differ only in the third base. There are 61 codons specifying 20 amino acids. Minimally 31 tRNAs are required for translation, not counting the tRNA that goes for chain initiation. Mammalian cells produce more than 150 tRNAs. Now, most RNAs have secondary structure consisting of stem and loop domains. Double helical states arise from base pairing between complementary stretches of bases within the same strand. Loops occur when lack of complementarity or the presence of modified bases prevents base pairing. Now, the cloverleaf model of tRNA secondary structure emphasizes the two major types of secondary structures, stem and loop domains. tRNAs typically include many modified bases and in the loop domains. Tertiary structure depends on interactions of bases at more distant sites. So many of these interactions involve non-standard base pairing or interactions including 3 or more bases. tRNAs usually fold in the L-shaped tertiary structure. By extending out from the receptor stem, the 3' end of every tRNA has a sequence CCA. Now the appropriate amino acid is attached to the ribose of the terminal A in the 3' end. The anticodon loop is the opposite end of the L shape. Non-standard H-bond interactions, some leaking 3 bases help stabilize the L-shaped tertiary structure of tRNA. Some other RNAs, including viral RNAs and segments of ribosomal RNAs, fold in pseudoknots, tertiary structures that mimic the 3D structures of tRNA. Pseudoknots are stabilized by tertiary non-standard H-bond interactions. Amino acid tRNA synthetases catalyze linkage of the appropriate amino acid to each tRNA. The reaction occurs in 2 steps. In step 1, an O atom of the amino acid alpha carboxyl attacks the P atom of the initial phosphate of ATP. In step 2, the 2' or the 3' OH of the terminal adenosine of tRNA attacks the amino acid carbonyl C atom. This is summary of the two-step reaction. Amino acid plus ATP, amino acid AMP plus PTI, amino acid AMP plus tRNA, amino acid tRNA and AMP. The two-step reaction is spontaneous overall because the concentration of PTI is kept low by hydrolysis at lysis of pyrophosphatase. Now this is there is a different amino acid tRNA synthesis for each amino acid. Each amino acid synthesis recognizes particular amino acid and the tRNA is coding for that amino acid. Accurate translation of the genetic code depends on the attachment of each amino acid to an appropriate tRNA, and domains of tRNA recognized by an AARS are called identity elements. So most identity elements are in the acceptor stem and the anticodon loop. So amino acid tRNA synthesis arose early in evolution. The earliest AARS probably recognized tRNAs only by their acceptor stems. Now there are two families of amino acid tRNA synthesis, class 1 and class 2. Two different ancestral proteins evolved into the two classes of AARS enzymes which differ in the architecture of the active site domains. They bind to the opposite sides of the tRNA acceptor stem, resulting in amino acylation of a different OH of the tRNA, 2' or 3'. Here the class 1 ARS identity elements usually include residues of the anticodon loop and accept the stem. Class 1 ARS amino acid link the 2' OH of adenosine and 3' end. Class 2 ARS identity elements for some class 2 enzymes do not include the anticodon domain. Class 2 ARS type 2 amino acid link the 3' OH of the adenosine and 3' end. Some amino acid tRNA synthetases are known to have separate catalytic sites that release by hydrolysis inappropriate amino acids that are misacylated or mistransferred to tRNA. The amino acid tRNA synthetase for isoleucine a small percentage of the time activates the closely related amino acid valine to valine AMP. After valine is transferred to tRNA isoleucine to form valine tRNA isoleucine, this is removed by hydrolysis at the separate active site of the isoleucine RNA synthetase. It accommodates valine but not at the larger isoleucine. So in some bacteria, editing of some misacylated tRNAs is carried out with separate proteins that may be evolutionary precursors to editing domains of amino acid tRNA synthetases. Some amino acids are modified after being linked to tRNA. Example, in prokaryotes and in mitochondria, initiated tRNA F-met is first charged with methionine. Methionine tRNA formyl transferase and catalyzes formation of the methionine moiety, usually THF, tetrahydrofuran as the formyl donor, to yield F-met tRNA F-met. In some prokaryotes, a non-discriminating ARS loads exported into tRNA AS and the exported moiety is then covered by aminotransferase to exclusion, yielding ASN tRNA AS. So glue tRNA glue GLN similarly formed and converted to GLN, tRNA, GLN in such organisms. Now, other roles of amino acid tRNA synthetases. In some organisms, amino acid tRNA synthetases are evolved to take on signaling roles in addition to the catalytic role of joining an amino acid to the direct tRNA. Examples have been identified of particular ARSs that regulate transcription, translation, or intrasplicing to binding to DNA or RNA. Proteolytic cleavage of human ARS for tyrosine and tryptophan produces active cytokines that regulate IgG. Several mammalian amino acid tRNA synthetases associate with other proteins to form large macromolecular complexes whose roles are actively being investigated. Eukaryotic cytoplasmic ribosomes are larger and more complex than prokaryotic ribosomes. Mitochondrial and chloroplast ribosomes differ from both the examples shown. Structures of large and small subunits of bacterial and eukaryotic ribosomes have been determined by X-ray crystallography and cryo-EM for the image reconstruction. Consistent with predicted base pairing, extracrystalline structures indicate that ribosomal RNAs have extensive secondary structure. This is a cutaway view at orange shows the tRNA. Okay, I'll not go into much detail. Large ribosomal subunit, active site, all of that. Synthesis. Now, the fundamental process of photosynthesis is the formation of a peptide bond between the carboxyl group of amino acid at the end of the polypeptide chain and a free amino group on another amino acid. So, proteins are made up from a set of 20 amino acids called standard amino acids, and each of the 20 amino acids is associated with specific codons. Now, one additional amino acid, cysteine, present in some polypeptide is directed by a modified reading of the genetic code, right? Polypeptide synthesis proceeds from N terminus to C terminus, and ribosome reads mRNA in the 5' to 3' direction. So, three kinds of RNAs RNA molecules perform different but cooperative functions in proteins. Okay. So we have read all of these. We will not go into too much detail. Not going into too much detail. Now, the first order translation is the binding of tRNA molecules to their appropriate amino acids called tRNA charging. Now, each tRNA is specific for a particular amino acid. All tRNAs have the sequence 5 prime, CCA 3 prime, right? So the 3 prime end and the carboxyl group of the amino acid is attached to the adenine nucleotide of the 3 prime end of the tRNA. So if each tRNA is specific for a particular amino acid, but all amino acids are attached to the same nucleotide at the 3 prime end of tRNA, how does the tRNA link up with this appropriate amino acid? So the key to specificity between an amino acid and its tRNA is a set of enzymes called amino acid tRNA synthetases. A cell has 20 different amino acid tRNA synthetases, one for each of the 20 amino acids. So each synthetase recognizes a particular amino acid as well as all the tRNAs that accept that amino acid. Recognition of the appropriate amino acid by synthetase is based on different sizes, charges, and R groups of the amino acids. The recognition of tRNAs by synthetase depends on the differing nucleotide sequences of the tRNAs, that is, the anticodon arm and the sequences in the acceptor arm. This schematic shows the charging of a tRNA molecule to produce valine tRNA. If you can see, the amino acid valine is amino acid and the ATP bind to enzyme. Enzyme catalyzes coupling of amino acid to AMP to form amino acid AMP. Two phosphates are lost in the reaction. Uncharged tRNA binds to the enzyme. Enzyme transfers amino acid from amino acid AMP to tRNA to form amino acid tRNA. The amino acid tRNA and AMP are released from the enzyme. Adding an amino acid to tRNA. The correct amino acid is attached to the tRNA by an enzyme called amino acid tRNA synthetase. The process is called amino acylation or charging and produces an amino acid tRNA. Amino acylation uses energy from ATP hydrolysis. First, the amino acid and the ATP bind to the specific amino acid tRNA synthetase enzyme. The enzyme that catalyzes the reaction in which the ATP is hydrolyzed to AMP, which charges the amino acid as AMP to form amino acid AMP. Next, the tRNA molecule binds to the enzyme which transfers the amino acid from the amino acid AMP to the tRNA that displaces the AMP. The enzyme then releases the amino acid tRNA molecule. Chemically, the amino acid attaches at the end of the tRNA by a covalent linkage between the carboxyl group of the amino acid and the OH group of the ribose of the adenine nucleotide found at the end of every tRNA. So, amino acid plus tRNA plus ATP, amino acid tRNA synthesis, amino acid tRNA, AMP plus UPI. Systematic of amino acid attachment to a tRNA molecule. We will not go into too much detail. Already had this discussion in a lot of previous classes. This is too much detail. Okay, I will not go into this much detail. Coming back to the question that we started with, for participation in protein synthesis, the tRNA must get amino acid. This was the last question for today. If you have any doubts regarding this question, please let me know now. I'll wait for a few minutes and then we'll conclude the session. If anybody has any doubt, please let me know.
Okay, so we can conclude the session for today over here. I'll meet you all. This was the last